Hi everyone, my name is Haley Garcia and I will be discussing the article titled Young Adults with Autism Spectrum Disorder Show Atypical Neural Activity During Emotional Face Processing. Autism spectrum disorder, or ASD, is characterized by a wide variation of signs and symptoms, and severity of those signs and symptoms, hence being a spectrum disorder. Difficulties in social interaction and communication, verbally and/or non-verbally, is a predominant feature, thus making it hard for individuals with ASD to function well in school, work, and other important areas of life. It is typically diagnosed within the first 2 years of life as symptoms begin to show in early childhood, but can be diagnosed at any age. The ability to socially interact and communicate well with others depends on being able to interpret nonverbal cues like facial expressions and to understand the emotional intent. Because individuals with ASD have difficulties in social functioning, it is presumed that emotional face processing is impaired. And here are some of the signs of autism. They may prefer to be alone, reject physical comfort, or have delayed speech development. Individuals with ASD lack interest in the human face and automaticity in face processing that occurs in normal development. It is also suggested that they don't find positive faces to be socially rewarding as typically developing individuals do. These findings may implicate in a poor development of affective processing, consequently leading to social skills deficits. Some studies showed intact affective processing, while others showed significant impairment, and this inconsistency may be due to different sample demographics, like age, task demands, for example, how long the stimulus is presented for, and the dependent variables chosen. Most previous research used fMRI or EEG neuroimaging, which don't achieve both good spatial and temporal resolution. fMRI studies showed atypical amygdala and fusiform activation, while EEG studies showed slowed or reduced neural responses, or similar responses to different emotions, indicating no emotion-specific activation. More specifically, there were no differences between fear and anger in youth with ASD compared to control group. This study used MEG, which, unlike fMRI and EEG, can afford both high spatial and temporal resolution, which is important for studying face processing. Studies on emotional face processing usually focus on neural responses to fearful faces, yet we encounter anger more often than fear. Both emotions are sources of threat, yet the responses to them are different. A person who looks fearful may promote altruistic, inclined to help type of behavior, whereas a person who looks angry may promote avoidance behavior to avoid the possibility of being harmed. Individuals with ASD have an atypical processing of angry emotional stimuli, and this processing is said to change with age. To further elaborate on that result, this study focused on neural responses to angry faces in young adults with ASD while yielding high spatio-temporal results and is the first study to do so. The researchers hypothesized that individuals with ASD have reduced and delayed activity during emotional face processing based on the reported deficits. 52 young adults participated in this study and were 19 to 36 years old. 26 had ASD and 26 were controls. To diagnose participants with ASD, the Autistic Diagnostic Observation Schedule for adults with fluent speech was used. Individuals were excluded if they had neurological or neurodevelopmental disorders other than ASD in the clinical group, used psychotropic medications in the control group, had any brain injuries, or were subjected to contraindications to MEG and MRI. There were no significant differences in IQ and accuracy and response latency for task performance between groups. Participants laid supine in the MEG while the stimuli were presented on a screen in front of them. A central fixation cross was first presented, then a happy, angry, or neutral face was presented with a scrambled pattern of the same face on the left or right side of the cross. Each participant had to indicate which side the scrambled pattern was relative to the cross by using a response button. Stimuli were presented for 80 milliseconds, and an interstimulus for 1300 to 1500 milliseconds. Participants completed a total of 300 trials, 50 trials for each expression in each hemifield, and each face was presented twice in each hemifield. Also, they completed practice trials to ensure that they understood the task. To accurately localize the source of activity, an MRI image was taken for each participant and used as an anatomical underlay for MEG. Pre-processing and analyzing MEG data involved statistical parametric mapping 12, and the empirical Bayes beamformer was used to localize sources of MEG activity. One of the major results was the early increased and prolonged activation of the right anterior insula in ASD in response to both angry and happy faces, which is shown in these two figures. Those with ASD are represented by the blue curve, while controls are represented by the yellow curve. You can see that over time, individuals with ASD had a greater activation in the AI. Now this figure displays neural activation in response to only angry faces, the length in milliseconds, and specific regions of over or under activation during processing of angry faces are shown. The blue bars represent regions of significantly greater activity in adolescents with ASD compared to controls, and the yellow bars represent reduced activation in adolescents with ASD. So you can see that this blue bar represents overactivation in the right anterior insula in those with ASD. This part of the brain is located deep within the lateral sulcus and is involved in subjective emotional experience. Salience, attention, empathy, and emotion processing are key functions. The right AI is structurally connected to the anterior cingulate cortex and amygdala, which are both key emotional processing areas, as well as to the nucleus accumbens, which plays a role in reward processing. Because there is atypical activity in the AI, which is connected to key areas involved in emotional and reward processing, it may attribute to aberrant functioning in deriving social reward from positive faces. Because the AI is also responsible for salience, atypical AI supports the notion that individuals with ASD have difficulty in attributing salience to faces. Early overactivation in the AI has been previously shown in those with ASD, and a 2013 study has shown a direct link between increased neural connectivity and increased severity in social deficits in ASD. Results also showed that young adults with ASD had no distinct neural activity when exposed to angry versus happy faces. This figure depicts greater activation in response to angry faces than happy faces within controls. These areas included the right inferior occipital gyrus, the calcarine sulcus, and the post-central gyrus. Previous results showed lack of emotional specific neural activity in young adults with ASD, which was mentioned before. And results from this study are concordant with a 2007 study which showed that different levels of fearfulness expressed in the human face elicited different neural responses in controls, but not individuals with ASD. There was also greater inferior temporal gyrus and fusiform gyrus activity in response to both faces. This figure only displays responses to happy faces, but you can see an increased activation in left and right areas of ITG and FG circled in red. ITG and FG have similar functions. Both are responsible for visual processing, object recognition, and facial identity. Consequently, this atypical overactivity may impair effective processing, considering this affects facial recognition and recognition of facial expressions. Results showed greater activity in the fusiform gyrus, which was unexpected, as previous fMRI results showed reduced activity, but these results were still atypical. There was also a laterality shift in overactivation such that there was overactivity in the left fusiform gyrus followed by the right, and similarly overactivity in the left ITG was followed by the right ITG. This could imply less automaticity during emotional phase processing, because greater activity could indicate a longer processing of object or facial features. Concordant with previous studies, there was an overactivation in the amygdala, specifically the right amygdala, in response to angry faces. This figure shows exactly that. The right amygdala showed greater activation in the ASD group compared to the control group. Amygdala is well known for processing emotions, especially fear, in response to threat stimuli. A 2004 study suggested that in normal development, subcortical systems involving the amygdala mature earlier and become less involved or active as you age during emotional processing, whereas frontal cortical systems like the anterior cingulate cortex mature later and become more involved. Therefore, this result of late increased activation in the right amygdala and decreased activation in the left ACC suggests an immature threat processing system. It is presumed that individuals with ASD don't respond to threat stimuli similarly to individuals without ASD. This is the first MEG study on emotional face processing in adolescents with ASD, which acquired results with high spatiotemporal resolution. Although results showed activation in areas that are known to be responsible for emotional face processing, activation was atypical. Overactivation was observed in the anterior insula, inferior temporal gyrus, fusiform gyrus, and amygdala. There were no distinct responses between emotional faces in young adults with ASD. To conclude, atypical activation during affective processing may influence the ability to derive social reward from positive faces, identify salience in faces, and influence the underdevelopment of the threat processing system, which consequently underlie social deficits in ASD. Lack of emotion-specific activation and overactivation in the amygdala has been supported by a lot of previous research, and further explains an immature threat processing system and the inability to derive social reward in faces. It is also important to consider that there was hypoactivation in the anterior insula in a meta-analysis of fMRI studies, but they focused on social processes beyond emotional face processing. A 2015 meta-analysis of fMRI studies found neither over or under activation of the anterior insula in ASD, and these differences may be due to different neuroimaging modality. Previous fMRI research has also presented reduced fusiform activity, which suggested dysfunctional face processing, but this study's results were the opposite. Therefore, more research is needed to define anterior insula and fusiform activity. Some of the questions I wanted to ask were about the diagnosis of ASD, would results be similar or different in children, and how would they want to further characterize these atypical patterns in future research? Here is my reference list, and thank you for listening to my presentation.
Today I will be discussing the paper, Time Course of Focused Ultrasound Effects on Beta Amyloid Plaque Pathology in the Transgenic CRND8 Mouse Model of Alzheimer's Disease, published in the journal Scientific Reports, issued by Nature Research. To start off, I'll go through the background of the paper, then some methods, the key results, conclusions, and then the possible directions of this research. The formation of beta amyloid plaques in the brain between neurons is thought to be a main indicator of Alzheimer's disease. However, when trying to eliminate these plaques, a main obstacle is the blood-brain barrier that prevents most molecules from passing through to the brain tissue. One way hypothesized to circumvent the blood-brain barrier is to use focused ultrasound, which is a non-invasive surgical treatment that has the ability to target specific areas of the brain. Microbubbles are commonly used along with this type of ultrasound because they can change in size in response to the ultrasound energy to increase the blood-brain barrier permeability. This study used Alzheimer's disease mouse models to address the questions of how a single focused ultrasound treatment affects beta amyloid plaques in the brain and how these effects of treatment change over time, which would allow an optimal treatment interval to be established. Some previous studies have already explored the effects of focused ultrasound on blood-brain barrier permeability, such as Burgess et al. That used transgenic CRND8 mice models and found them to have less permeable vasculature compared to the non-transgenic counterparts. Also looking at Alzheimer's, Liu et al. Compared the additional treatment of focused ultrasound with BAM10, a beta amyloid antibody, to a regular sialoinositol treatment. However, they found no difference in plaque reduction with the additional treatment. Looking at its application to other neurodegenerative diseases, Zhang et al. studied focused ultrasound microbubble treatment with an anti-alpha-synuclein antibody as a therapy for Parkinson's disease, which saw a reduction in alpha-synuclein. Even though a considerable amount of literature can be found on similar focused ultrasound studies, there have yet to be any discussing the questions that this study has addressed. A few questions left unanswered are those about which added pharmacological treatments could be used to optimize disease outcome, as well as what those outcomes might look like. Additionally, most studies have looked at advanced disease mouse models, which leads to questions about how focused ultrasound treatments could possibly help those exhibiting early-stage Alzheimer's. Right now, the important discovery of an effective Alzheimer's disease treatment in mice will lay the foundation for a human treatment to be developed, possibly answering some of the previous questions. Furthermore, exploring the treatment effects on biological mechanisms would provide important information for future scientific discovery, and being able to assess the optimal treatment course will lead to more advanced understanding of the treatment itself and how it might impact humans in the future. To resolve the first question, 2-photon microscopy was used on 9 6-month-old transgenic CRND8 mice that were split into a treatment group and a control group. Beta amyloid plaques were then mapped using adjacent plaque-covered vessels, using images taken at multiple laser powers to correct for the interference from the leakage of dextrin used for visualization. Microbubble solution was given to both groups, but the control group had the ultrasound transducer removed to prevent treatment administration. To examine the second question, magnetic resonance-guided focused ultrasound was used to target the bilateral hippocampi once every 2 weeks using 42 male and 7 female mice at 7 months old. The transgenic mice were separated into treatment and control groups, and the same for non-transgenic mice. Microbubble solutions were given to all groups, and images were collected for targeting and permeability assessment before and after sonication, which was then followed by analysis. There were 36 plaques mapped in the control group with 18 measured in the treated group. Results shown here in Figure 1 shows the change in plaque volumes and cross-sectional area with respect to time in treated and control mice. As you can see, beta amyloid plaques were reduced in treated mice after 2 days, but the largest cross-section did not increase until the 3rd week. The significant reduction after treatment continued to be significant until the 14th day, but volume increased slowly from day 2, remaining below the previous maximum until day 21, where it then surpassed the original value. When plaques were grouped based on size, smaller and larger plaques had similar reduction rates and growth rates. Throughout the 10 weeks that the experiment was being conducted, 7 mice died, but there was no mortality significance between genotypes. Here, Figure 2 shows that there is a significant difference in the average number and size of beta amyloid plaques after biweekly treatment between treated and controlled transgenic mice. However, it was found that maximum cross-sectional area did not have a significant change. There was also seen to be a greater reduction in beta amyloid observed in older mice that exhibited a further disease progression. Regarding permeability, localized dextrin flow was observed, which supports the permeability of the blood-brain barrier and specificity of the ultrasound target. Additional results showed no difference in permeability between transgenic and non-transgenic mice. This study concluded that one focused ultrasound treatment can reduce beta amyloid plaques starting the 2nd day until the 14th day, with biweekly treatments producing an additive effect. This identifies how the treatment affects beta amyloid, as well as how it changes over time. A further conclusion about the effects on beta amyloid itself were also found, such as that biweekly treatments were just as effective as weekly. This was evident by the exhibited decreases in biweekly versus weekly not being significantly different from one another. It was also concluded using a highly specific beta amyloid probe that plaque decrease was due to reduction in plaques already in existence and not just the inhibition of further growth. In contrast to previous literature, no difference was seen between transgenic and non-transgenic blood-brain barrier permeability, which signifies that Alzheimer's disease does not cause the increased permeability. Furthermore, control and treated mice had plaque growth after the 3rd week, which excluded effects from the cranial window insertion. The results, however, likely resembled a reduced effect of focused ultrasound treatment, since many of the treated mice did not receive all 5 scheduled treatments due to trouble with inserting the tail vein catheter. These results and conclusions align with previous literature, and that one study found a decrease in beta amyloid in mice models after sonication, while another also observed a decrease in beta amyloid but in rabbit models. While there are no major criticisms about this paper, any remaining doubts can be resolved by future directions of the research. This would include further investigation of the biological mechanisms of beta amyloid plaque production, such as the role of pro-inflammatory cytokines or microglial and astrocyte activation that has previously been reported to increase after focused ultrasound treatment. Alternatively, beta amyloid aggregation suppression could be caused by focused ultrasound treatment, which means further studies regarding this mechanism could be useful. Conjunction treatments are also a possible direction that have been previously explored, such as the use of BACE1 inhibitors to prevent beta amyloid production, or beta amyloid antibody injections alongside the ultrasound treatment. Like many of the preceding studies, this was conducted for a relatively short period, so more long-term effects should be examined to determine side effects or changes in plaques after the continuous usage. For instance, a decrease in blood-brain barrier sensitivity to the focused ultrasound treatment could be a result after an extended amount of time. Another direction could also be a study involving the measurement of behavior changes after the beta amyloid reduction. Since Alzheimer's disease symptoms include cognitive deficits and memory loss, it would be useful to know whether any observed symptoms would also be reduced, or whether this treatment would just slow or stop any further impairments. As mentioned before, a question I have about the study is whether the findings could apply to early-stage Alzheimer's disease, or even as a way to prevent progression. Understanding that future studies would be needed to address this question, in theory, would the use of this treatment earlier in disease progression lead to a prevention of the severe form of Alzheimer's disease? This kind of leads to another question about what the end goal of this research is. For instance, is it aimed at finding a cure, or is it more suitable as a maintenance method? If this treatment proved effective for humans, how might this affect those diagnosed with Alzheimer's? Would it be a widespread treatment? Would it be affordable? And would everyone be eligible to receive such treatment? That concludes my presentation. If there are any questions, feel free to reach out to me. Thank you so much for your time.
Hi, my name is Spandan, and today we'll be talking about this paper by Arjun Balachandran and Stephen A. Prescott. It was published in the Journal of Physiology in 2018, and it's titled The Origin of Heterogeneous Spiking Patterns from Continuously Distributed Ion Channel Densities, a computational study in spinal dorsal horn neurons. There is obviously a lot to unpack here, so let's get right to it. Neurons are often classified by their spiking patterns, and this is often assumed to be due to qualitative differences in the ion channels on them. That is, neurons with different kinds of ion channels on them would be expected to spike differently. However, quantitative differences in ion channels, that is, how dense each channel is on a neuron, can also produce different spiking patterns if they operate near what is called a bifurcation, where a small change in the continuous variation of a parameter causes a discrete or discontinuous change in the output. So, slight changes in ion channel density or the test conditions under which we observe the neuron may cause qualitatively different spiking patterns if it operates near a bifurcation. Now, the neurons we're talking about here are superficial dorsal horn neurons. These are present in the lamina 1 and 2 of the spinal cord and are involved in the early processing of somatosensory information, especially thermal and nociceptive input. Now, most of the SDH neurons are observed to be local interneurons, with a third of them being inhibitory while the others are excitatory. The spiking patterns in SDH neurons are noted to have been sensitive to stimulus intensity as well as the pre-stimulus membrane potential, indicating that maybe they're operating near a bifurcation. The authors of this paper have hypothesized that the heterogeneity of spiking patterns in an entire population of neurons occurs due to the distribution of ion channel densities being straddling across a bifurcation. That is, there are different neurons in the population that are on different sides of a bifurcation, and as a result, they spike differently. Using a computational model, the authors covaried the densities of two different potassium channels to map out where exactly these bifurcations would occur in the parameter space of these densities. The channels that they looked at were a low threshold non-inactivating potassium channel of the Kv1 type, as well as an inactivating potassium channel of the A type. So these recordings were taken from rat lamina 1 neurons, and they indicate the different spiking patterns that can be observed in SDH neurons. So the different patterns can be grouped into 6 major categories. Tonic, which is a neuron spiking at regular intervals. Phasic, where it fires in phases, so it fires rapidly and then does not fire. Delayed, when there is a delay before the first spike compared to the intervals between spikes. Gap, where the first spike and the second spike is separated by a larger time interval compared to the other spikes. Single, when there is just a single spike that occurs followed by no other spikes, and reluctant, when the cell does not fire at all. So in part A, all the recordings here were taken from different cells, but in parts B, C, and D of this diagram, we see the recordings taken from a same cell under different conditions, showing how the same cell can fire in different spiking patterns depending on the test conditions. So we see that in part B, a cell can go from tonic to phasic to even single spiking depending on the stimulus intensity that is applied to it. Now, the methods that the authors used, obviously this is a computational study and there has to be a model of a neuron made. Now, the model they used was a modification on what's called the Morris-Lecar model. Now, the original model only allows like 3 conductances. Now, these conductances correspond to the fast activating sodium channels, the delayed rectifying potassium channels, and the leak channels. The authors added 2 more conductances to this model corresponding to the 2 potassium channels of interest. So the one highlighted in red corresponds to the low threshold non-inactivating channels, while the one highlighted in blue corresponds to the A-type inactivating channels. So that gives us this resultant cable equation, which is essentially just a conservation of charge equation, and it tells you how the voltage on the neuron varies in time. The second equation, it essentially describes how the gating variables W, Z, A, and B change with respect to time. There are a bunch of other parameters involved here, and they are described on this slide. The values for a lot of the constants were taken from the literature, and they are mentioned in the paper. Now, what the authors did was they varied the 2 channel conductances in a systematic manner, and they simulated the neuron to reach a steady state before a stimulating current was applied to it. The resultant spiking pattern was then observed and classified. The same model was run for different values of the stimulus intensity in order to constrain the channel distribution for a given sequence of spiking patterns, something I will get to a little later. In a population of neurons, the ion channel densities of the 2 channels were assumed to be normally distributed. Now, these 2 normal distributions were combined along with the coefficient of correlation between them to produce a bivariate normal distribution. The bivariate normal distribution would have a probability density function across the entire parameter space, and integrating over the entire parameter space should give you a value equal to 1. Now, to find the proportion of neurons in each region of the parameter space corresponding to a specific spiking pattern, we would do that by integrating the probability density function of the bivariate normal distribution over that region specifically. Moving on to the results, the first step was to try and reproduce the spiking patterns we observed in the rat neurons. So starting off with the model without the 2 channels of interest, so a model that only has a fast activating sodium channel, a delayed rectifying potassium channel, and the leak channel, and using a stimulus intensity of 60 microamperes per square centimeter, we see that the neuron fires in a regime of tonic spiking. However, if we add the late rectifying non-inactivating potassium channel, we see the neuron goes into single spiking. This occurs because the first spike can go off before the channel activates. However, after the channel activates, the cell becomes hyperpolarized and no more spikes are possible. Going back to the original model and adding the A-type inactivating potassium channels, we see that delayed spiking is observed. This happens because the first spike is prevented because of the channel activating, but then subsequent spikes can take place after the channel inactivates. If these channels are present in low enough density, then we see gap spiking. This happens because the density of the channels is not high enough to prevent the first spike from taking place right at the start, but after it does activate, spikes cannot take place. So subsequent spikes can only take place after the channel has been activated. If both these channels are present in a neuron, we see reluctant spiking, where the neuron basically does not spike. The first spike is prevented due to the A-type inactivating channels, and any subsequent spikes are also prevented because of the late rectifying non-inactivating channel. Here we see that the two channel densities were varied systematically to create a parameter space, with each channel density on either axis. Now the regions in the parameter space represent any combination of the channel densities which would result in a certain spiking pattern. Within a certain region, any of the combinations would produce the same spiking pattern. So any neurons having channel densities corresponding to the green region would display delayed spiking, as you can see between neurons A and B, even though they have pretty dissimilar combinations of channel densities. However, if two neurons are on either side of a boundary, they would exhibit different spiking patterns, even though the channel densities would not be that different between them. For example, A and D are pretty close in terms of the channel density of the um, low threshold non-inactivating channel. However, neuron A exhibits delayed spiking while neuron D exhibits reluctant spiking. On the right, we see how changing the test conditions can cause these boundaries to shift. For example, on the top picture, we see that changing the stimulus intensity causes the boundaries to shift, and as a result, the same neuron can exhibit different spiking patterns at different stimulus intensities. Below, we see that the same occurs with changing the pre-stimulus membrane potential. So why is this important? By looking at the sequence of spiking patterns across a variety of different test conditions, we can constrain the estimate for the ion channel densities present in that neuron. For example, if we look at neuron A here, we test it at a variety of different stimulus intensities going from 50 microamp per square centimeter to 80 microamp per square centimeter. We see what kind of sequence of spiking patterns it shows, and as a result, we can say that it definitely has an ion channel density that is constrained within that gray rectangle, because those are the only densities that can produce this particular sequence of spiking patterns. We see the same for neuron B. However, the constraint is much weaker, so we cannot have as tight an estimate for neuron B. Essentially, as close as the neuron is to bifurcations, the more tightly we can constrain its estimate. Moving on to the population picture here. Here we see the two univariate normal distributions for the channel across a population combined to give the color map, which represents the probability distribution of the joint normal distribution. Here, the hotter the color, the more probable it is to find a neuron with that particular combination of channel densities. If the two univariate distributions have equal width, then we see that the joint distribution appears circular. However, if the widths are different, as you see in part B, it takes the form of an ellipse. In both A and B, the two univariate distributions have no correlation between them. However, if there is a correlation, it creates a slant in the ellipse. If the correlation is positive, the slope of the long axis of the ellipse is positive, and if it's negative, the slope of the long axis becomes negative. What is this useful for? So we know that the entire probability distribution, if we integrate it over the entire parameter space, it would give us a value of 1. So therefore, if we integrate the portion of the probability distribution that lies over a certain region of the parameter space, we can say what proportion of the neurons would be firing in that particular spiking pattern. What the authors are trying to do here is to take a population of neurons whose channel density distribution they know. Now they're trying to figure out what the proportions of the spiking patterns are in that population. Now we try to use an algorithm to find an estimate for the joint distribution of ion channel densities that would agree with the proportions of spiking patterns ratio. And obviously now we have something to compare it with to know if it's really working. How does the algorithm work? First, we start off with an arbitrary distribution with two means corresponding to the two univariate distributions with no correlation between them. Next, we try and optimize the correlation coefficient so as to minimize the error between the estimate and the true proportions of the spiking patterns. Next, we try and optimize the two means. Once this is done, we go back and try to optimize the correlation again. And this is done iteratively over and over again till the final spiking pattern proportions that are estimated by the algorithm are close enough to the true values. So what are the major takeaways from this paper? The authors were able to reproduce 5 of the spiking patterns observed in SDH neurons by only varying the density of 2 channels. Next, there are boundaries in the parameter space of the channel densities corresponding to bifurcations, and subtle changes to the channel densities can cause a change in spiking pattern if it crosses a certain bifurcation. However, the regions of the parameter space associated with each spiking pattern are pretty large, meaning a wide range of channel densities can cause the same spiking pattern. Next, we saw that functional heterogeneity in a population of neurons, that is, in the same population of neurons, different neurons spiking with different spiking patterns, do not reflect cells with different channel types, but can be explained by continuous variation of channel densities, that is, cells having different densities of the same channels. Now, why are these findings important? First of all, these results are concordant with previously observed temperature-dependent switching between delayed and gap spiking in these neurons. Now, this can be explained by the fact that delayed and gap spiking lie on either side of a bifurcation, and a small change in the test conditions, as can occur due to a temperature change, can cause this boundary to shift, and a neuron that was originally delayed spiking can have crossed the boundary and is currently gap spiking. Furthermore, GABAergic and glycinergic neurons were found to be almost exclusively tonic spiking, while glutamatergic neurons exhibited a far more heterogeneous spiking pattern. Also, like inhibitory neurons, which express parvalbumin, did not show any of the spiking patterns that the excitatory neurons showed. This could perhaps be an indication that tonic spiking neurons are inhibitory, while the ones that spike with delayed single gap or reluctant patterns are excitatory. This does not mean that excitatory neurons come in 4 different types. They just mean that they have different densities of the same channels, which cause the different spiking patterns. Finally, where can we go from here? Although the authors used a method to estimate the channel density distributions, and it works in principle, it does have a few limitations. Firstly, the standard deviation of the estimated distribution has to be assumed to be equal to the true distribution. Otherwise, it not only induces a lot more error, but also makes the computation much longer. It also does not involve other factors that could be involved in the heterogeneity of a population, such as other channels that might be involved. To incorporate all of this would require a model that would not work in the two-dimensional parameter space that we saw, but in a multi-dimensional parameter space where the distribution would have to be represented as a manifold. This would not only be much more intensive computationally, but also require much more careful analysis, as correlations would have to be pairwise and multiple different interactions would have to be considered. Also, functional heterogeneity based on the correlation of different channels is something that has not been studied in detail yet, and as the authors say, it definitely deserves a look in the future. I would like to end this by saying a big thank you to Dr. Bill Ju and the entire HMB 420 team. It's been an amazing course, and I'm really grateful for having been able to take it. If you've been sticking with me for this long, and you want a little TLDR on the entire paper, the screenshot on the right does a decent job at summarizing the whole thing. It's a pretty dumbed down version, but I'm pretty proud of that explanation. So with that, thank you. It's been a great time.
Hello, my name is Jeanette Titus, and I'm a fourth-year undergraduate student majoring in neuroscience at the University of Toronto. Today, I will be presenting my journal club presentation based off of the research paper, Structural and Functional Consequences of Chronic Psychosocial Stress on the Microbiome in Host. Thank you, and I hope you find my presentation interesting. Here's the first page of the paper that I will be presenting. And if you're interested in reading the original paper, I have it cited in my resources page at the end of my presentation. Here's the outline for today. I will be first talking about the background and overview of the paper. Background and overview. The U.S. Department of Defense funded this research because they were interested in how probiotics may improve the outcome of individuals coming back from combat and reducing the symptoms of PTSD, as well as understanding the microbes that are involved in PTSD. The microbiota is a large collection of mutually beneficial bacteria that plays a role in gut homeostasis, the regulation of the gut immune system, barrier integrity, and nutrient absorption. Multiple animal studies suggest that stress can change the composition of the microbiota through inflammation, which has been shown to be implicated in many psychiatric diseases such as depression or PTSD, which is known as the gut-brain axis. The gut is often called your second brain. There is a long literature regarding the crosstalk that happens between the microbiota and the immune system, and how stress can modulate this due to lack of signals, and then create a lack of maturation of the gut microbiome. This paper used the chronic social defeat model in order to apply stress as a stimulus, and then view the behavior that followed. In previous literature, this has been shown to induce social anxiety and create a reduction in social behavior, which are common symptoms of PTSD. The hypothesis of the study is that chronic exposure to psychosocial stress impacts microbial markers, innate immunity, and biological pathways via structural shifts in the microbiome. These results are hypothesized to give long-lasting effects. The rationale is that inadequate coping behavior to psychosocial stress contributes to gastrointestinal and neurological disorders through the gut-brain axis. There is a long literature regarding the gut-brain axis and the role of the immune system in this pathway, which is why the authors created this experiment. Lastly, the relevance is that there is a growing importance of connection between the microbiota and development of metabolism, immunity, and social behavior. Information provided in this study will help the authors answer questions regarding stress, the microbiome, and PTSD. Many things are still unknown regarding the microbiota and the role it plays in the immune system, metabolism, and the development of psychiatric disorders, but there is enough information available for the researchers to try to answer these two questions I have outlined here. Next, I will talk about the materials and methods used in this study. Animals used in this study were a male C57 black 6 mice and a CD1 aggressor mice. They were able to acclimatize to conditions and were fed ad libitum. The social defeat that was used was a knockdown of BDNF. It was used for the development of social aversion and has similar effects to the administration of an antidepressant. This was conducted over 10 consecutive days. Behavioral tests that were conducted included an open field test, 3-chamber test, light-dark test, and aggressor interaction. The analysis done included splenocyte isolation, which occurred 5 or 17 days post the final defeat. Flow cytometry, where splenocytes were stained for markers of dendritic cell maturation, such as CD11, MHC2, CD80, and Tregs. 16S rRNA bacterial analysis was done via fecal pellet isolation. In silico metagenomics were performed, where predictions regarding the functional composition of the microbiome were made on specific OTUs. Finally, statistical analysis was also performed. Next, I will talk about the major results. For the first figure, the researchers focused on the effect of chronic social defeat on behavior. Defeated mice spent less time in the aggressor interaction zone and more time in the empty chamber, rather than the center or mouse chamber, where unstressed control mice spent more time, shown in the top two graphs. Defeated mice also had lower sociability scores, so they spent less time interacting with mice or an object in the chamber. In the open field test, defeated mice showed reduced locomotion and exploratory behavior. Because anxiety behavior was similar in the open field test between the two mice, anxiety was instead tested using the light-dark test, and here defeated mice made fewer entries into the light zone. Figure 2 focuses on the effects of stress on microbial diversity. The authors profiled the composition of mice that were under prolonged stress. There was an overall decrease in the diversity of the microbiome following stress, where the defeated mouse post-stress showed the largest decline, as well as a decline in the richness of the community shown in the top two graphs. Defeated mice showed lower levels of Akkermansia and Corticobacteriaceae, which are normally associated with healthy and stress-exposed communities. This figure looks at the effect of chronic social defeat on the microbiota and how it influences behavior. Researchers looked at the distribution of OTUs in the defeated and control mice. The abundance of firmicutes and Bacteroides, which are the major phyla, were similar, but when the class level was examined, differences were noted in the Bacilli and Clostridia species. Figure 3b shows 25 OTUs were shown to be represented differently between the two groups, but there is no clear trend shown, which showed how complex alterations can be following stress. Finally, Figure 3c showed a statistically significant trend towards the microbiome and avoidance behavior in stressed mice. Figure 4 looked at the long-lasting effects that chronic social defeat has on the immune response, due to the long literature focused on the communication between the immune system and the gut-brain axis. In defeated mice, IL-6 levels were increased 5 days post-stress, as well as IL-10 expressing CD4 positive and CD4 positive T cells, shown in the top two graphs. 17 days post-stressor, there is a decrease of the previously described T cells, as well as a decrease in the release of IL-10 from splenocytes of defeated mice. Dendritic cell markers were analyzed both 5 and 17 days post-stressor, and there is an increase found at both time points, which suggests enhanced dendritic cell activation and a role of the innate immune system. The final figure focuses on the predicted metagenomic profile that is expected post-chronic social defeat. The defeated group had a lower number of pathways involved in fatty acid metabolism and of propionate and butyrate, which are bases of short-chain fatty acids, propinic acid and butyric acid, as well as a reduction in pathways involved in tryptophan and tyrosine production. These are important because they serve as precursors for dopamine and 5-HT serotonin. In the following slides, I will go over the main conclusions from this paper. Number 1. Psychosocial stress induces OTU shifts in the structural composition of intestinal microbiota, as well as changes in abundance of the microbial groups. This was shown in Figure 3, where class differences were identified as well as differences in OTUs between the control and defeated mice. The major conclusion made was that following chronic social stress exposure, defeated mice showed distinct intestinal microbiota from the control group, and showed a more complex form of dysbiosis by identifying different OTU shifts. Second, social defeat reduces the prevalence of synthesis and metabolism pathways for short-chain fatty acids and neurotransmitter precursors. This showed how structural changes in the microbiome are associated with functional consequences on biological pathways that may influence behavior or have an impact on psychiatric diseases. Third, the alterations of intestinal microbiota show a connection between stress-related psychiatric disorders and functional bowel diseases. This relates back to the major question the researchers want to answer regarding PTSD, probiotics, and the microbiome. It was shown how aggressor avoidance in this study following social defeat bears a similarity to phobic avoidance behavior, which is shown in PTSD. Lastly, exposure to social defeat induced long-term alterations in the immune profile and innate immune system. As shown in Figure 4, there was differences in IL-10 levels, dendritic cell markers, T cells, and Tregs, which are all indicative of a sustained innate immune activation and long-term change. I was very fortunate to secure an interview with one of the authors of this paper, Dr. Jane Foster. She gave me a lot of insight into her paper and her research, and helped me understand the results and relevance of this paper. On this slide, you can see the questions I asked her, and the answers that she provided me I've incorporated into my discussion of the paper. Next, I will focus on the critical analysis and future directions. Critical analysis. First, this paper addresses a gap in the literature by providing functional context to structural microbiota changes using an adult stress model. This also helped branch the functional relationship between the microbiome and stress-induced changes that can impact the brain. The study also provided long-lasting results and effects of stress on the microbiome, immune system, and psychiatric diseases, as many other studies only provide short-term results. So this was a very good experiment to model PTSD, which is a long-term disease. Although the study utilized only one strain of mice and one gender, this can come with limitations to interpretation of results, since every strain of mice reacts differently to stress, and each strain has a specific microbiome profile. The study lacked focus on specific groups and number of bacteria thoroughly, which is mostly due to a lack of references available in the field. Dr. Foster emphasized that the field is changing very quickly, so if these experiments were done today, more references would be available to determine these differences. Another critical analysis I have is that individuals exposed to severe wartime stress have a greater risk of IBS, associated with alterations in the microbiota. It was shown here how long-lasting effects of stress can impact certain psychiatric conditions, and previous literature has outlined the importance of the immune gut-brain axis in these instances. This goes on to my second point of how exposure to social defeat induce alterations in immune system, which is observed in psychiatric conditions. This study did demonstrate how chronic social defeat and stress induces long-term changes in the immune regulation and priming of the innate immune response by the microbiota. Lastly, the findings did agree with previous literature regarding short-chain fatty acid synthesis and other metabolites that are impacted by the microbiome dysbiosis. For example, tryptophan is a precursor to 5-HT-serotonin, and more than 90% of it is synthesized in the gut by the microbiota. This is done by predominantly Clostridia, which induces IL-10. In this paper, defeated mice had a lower Clostridial species and a lower IL-10 production, therefore agreeing with previous literature on how short-chain fatty acids dysbiosis can lead to neurological disorders. Here I will talk about the future directions I have outlined for this paper. The first suggestion I have is that social defeat model used in the study should be applied to different strains of mice to determine if the effect is strain-specific or a general phenomenon. As I mentioned before, different strains have different microbial makeups and respond to stress differently. It would also be beneficial to use this stress model on female mice to determine if this is gender-specific as well. Next, I would incorporate new technology and reference databases from the microbiome field in order to grasp a better understanding of the specific changes in microbes. Because the gut microbiome field is changing fast, it would be useful to incorporate new technology and advancements into this experiment in order to grasp a deeper understanding of the changes that are occurring. I would also try to introduce a new stress model by incorporating chronic stress early in life to determine if results have long-lasting effects into adulthood. Because PTSD is a long-term condition, not short-term stress such as taking an exam, to determine if stress does impact in the long term, it should be applied early in life. It would be beneficial to apply the stress as soon as the rat is born, or possibly in utero, to determine if these have similar or different outcomes. Lastly, I would propose a new model involving knockout of specific immune cells such as T cells, which are known to be important in the gut immune crosstalk. It was mentioned in my interview that T cells are very important in delivering signals to the microbiome in order for it to develop. And if this doesn't happen, it can lead to a cascade of negative outcomes such as lack of protection, immune function, and homeostasis. This concludes the end of my presentation. Thank you so much for listening to me speak about the research that was done in this paper. If you have any questions, you are free to contact me. And again, thank you so much to Dr. Foster for all of her insight. Thanks again.
Okay. Hi, everybody. My name is Alex Vachon. The article I will be presenting on today is Altered Blood Biomarker Profiles in Athletes with a History of Repetitive Head Impacts. As you can see on the bottom right is my citation, and my person of interest is Andrew J. Baker. So moving on to just a brief explanation for those who may not understand all the terms in the title. So what are biomarkers? It's a great question. Biomarkers can be certain characteristic biological properties or molecules that can be identified and then measured in various compartments in the body, including blood or tissues. Biomarkers can be used to indicate normal or diseased biological processes in the body, including cancer cells. Biomarkers can also be distinct cell groups, molecules, genes, enzymes, or hormones. So moving on, another question, what is a concussion and why is it bad? A concussion is a mild traumatic brain injury that affects how your brain functions either short-term or long-term. This brain injury is due to the fact that when one receives a blow to the head, the impact causes the brain to move rapidly back and forth, kind of like in a rattling motion, inside your skull, damaging brain cells and or structures. These damaged brain cells and or structures can cause physical and chemical changes in your brain, which can have a resounding effect on behavior and cognitive ability. So now moving on to background and overview. So there is surfacing evidence arguing that both inflammation and neurodegeneration are crucial to secondary injury process and the root cause of neurodegenerative diseases. Current understanding of concussion pathophysiology comprises an acute disturbance of neurobehavioral function coupled with damage to neuronal and glial cells. Symptoms, usually short-lived, however, with recent advancements in neuroimaging and analytical biomarker assessment, underlying structural and functional abnormalities lasting long after symptom resolution have been documented. There's new evidence that suggests that concussions may contribute to negative health outcomes, including CTE. In this study, they aim to identify differences in the systemic biomarker profiles of male and female athletes who sustain multiple previous concussions, and in males specifically who participate in collision sports. In other words, the study aimed to characterize brain injury and inflammatory mediators in healthy male and female athletes according to concussion history and the participation in collision sports. So just a brief overview on inflammation. Inflammation is critical for both repair and neurodegenerative processes post-trauma. Resident microglial cells are important for the regeneration of damaged brain tissue through the release of neurotropic factors. However, chronic inflammation may impede neuronal and glial cell injury, resulting in further degeneration, which can lead to the deposition of neurofibrillary tangles and amyloid plaques. Therefore, there is a very, very fine line between benefit and exacerbate. For future reference, MCP4 is a human monocyte chemoattractant protein with an influence on monocytes, eosinophils, and basophils, which basically comprise a small part of the innate immune system. MCP1 is another monocyte chemoattractant protein with similar influences. So the reason I'm mentioning these now is because I'm going to allude to these later. These are very important part of this article because they're a part of the major finding that these individuals had. So moving on to materials and methods. So the participants were recruited from the University of Toronto varsity athletic teams between August 2014 and December 2015. Medical history of the athletes were obtained by the team's therapist and or trainer. There were 16 teams that were contacted for this study, 8 male and 8 female. Athletes were excluded if they suffered from seasonal allergies, cold, infection, inflammatory related conditions, had musculoskeletal injuries, already had concussive symptoms, or were taking any medication other than birth control. These procedures were approved by Health Sciences Research Ethics Board at the University of Toronto, and all athletes were provided written informed consent. A total of 87 athletes were included in this study, 60 of which were males and 27 females. All athletes were of similar age. Now to measures. What was used was the Sport Concussion Assessment Tool 3 with the acronym SCAT-3. SCAT-3 combines previously recognized concussion tools into 8 components designed to properly monitor or assess concussion symptoms. Each of these components are scored and recorded. The results are scored on a concussion system scale using a 7-point Likert scale rating. The severity of the symptoms are calculated by adding the rated symptom score for each symptom. Now on to blood collection. After written consent, venous blood was taken from athletes before their season started, like in the upcoming year. Venous blood samples were captured in a 10 milliliter K2 ETA tube. Within 1 hour of being drawn, samples were centrifuged for 15 minutes at 4 degrees Celsius. Subsequently, the plasma supernatant was separated and froze at -70°C until it was ready to be analyzed. So continuing in the materials and methods section, now onto the statistical analysis. So students' independent t-test, the Mann-Whitney U, and chi-squared tests were used to evaluate the demographic and descriptive statistics of both male and female athletes. Analysis of sports were divided between collision, which would be purposeful contact such as football, and non-collision sports. Individual biomarker values were excluded if they didn't meet manufacturer's recommended level of quantification for each analyte. 96 well plates were analyzed. Plates only included in analysis if variance between plates was less than 20%. Biomarkers were not included in multivariate analysis if greater than 30% of the data points were absent from any group. Multivariate analysis was performed using partial least squares discriminant with the acronym PLS-DA. The outcome is based solely on predictor variables and binary response variables. As an example, 1 would equal a correctly predicted outcome, 0 would equal an incorrectly predicted outcome. The partial squares least discriminant output provides model of p-prob. P-prob is the likelihood on the PLS model identifying the correct outcome conditional on the observed subject scores. In this study, covariance between the peripheral blood markers, which would be represented as predictor variables, and both concussion history and collision sport participation, which are represented as response variables, were assessed individually between athlete gender groups. Significant biomarker values were found by performing bootstrap resampling on subjects to obtain p-values which were corrected for multiple comparisons at a false discovery rate of 0.05. Now on to the last slide of materials and methods, I promise. Biomarker analysis. 28 out of 39 markers were analyzed using MSD 96 well multi-array spot V-plex human immunoassay kits. These were run on an MSD sector imager with Discovery Workbench software. An additional assessment panel assessed by multiplexed immunoassay was used to evaluate additional neuroinjury markers, including total GAU, GFAP, which stands for glial fibrillary acidic protein, S100B, which stands for S100 calcium binding protein B, NSE, which stands for neuron specific enolase, NRGN, which stands for neurogranin, CK-BB, which stands for creatine kinase BBB isoenzyme, VILIP-1, which stands for vicinin-like protein, VWF, which stands for von Willebrand factor, BDNF, which stands for brain-derived neurotropic factor, PRDX-6, which stands for peroxyrhodopsin, and monocyte chemoattractant protein MCP1, which I already mentioned. Now on to the results. So as for demographics and clinical characteristics, there were no significant differences in medical history and SCAT3 symptoms at the time of the study between gender groups. No significant differences between male and female athletes regarding age, concussion history, days since last concussion, number of previous concussions, and medical history. The only statistically significant value was that a higher proportion of male athletes played in collision sports as compared to female athletes, and this yielded a p-value of 0.001. Now on to the results associated with systemic inflammatory marker analysis. There were no significant differences between male and female athletes who did not participate in collision sports or did not have previous history of concussions. Now on to multivariate analysis of inflammatory markers. No individual biomarkers were significantly correlated to previous concussion history in either male or female athletes. Athletes with one previous concussion displayed no significant differences in biomarker levels when compared to athletes with no concussion history. This is where it gets interesting. When comparing athletes with multiple previous concussions to those with no previous concussions, female athletes had significantly higher MCP1 levels, whereas males had significantly higher MCP4 levels in plasma concentrations, encompassing all biomarkers according to concussion history. Only collision sport participation significantly covaried with an increase in tau. So if I can direct your attention to the top right of the slide, as you can see the bright fuchsia green. Bright fuchsia green represents the statistically significant data. As you can see in the brain injury markers, tau was statistically significant, as well as collision sport. So now moving on to what I like to call results explained, which is in part discussion. So the first finding was that female athletes with a history of multiple concussions had elevated blood MCP1 levels and males with the same exact history had elevated MCP4 levels. Chemokines, which is MCP1 and MCP4, are crucial for peripheral immune cell migration to the CNS post-injury. However, treatments involving the inhibition of chemokine recruitment have been successful in reducing cerebral damage and cognitive deficits in animals. It is difficult to speculate whether these findings suggest detrimental or reparative processes, as chemokines may be necessary for neuronal repair. While MCP1 and MCP4 are both involved in leukocyte recruitment, they may be dissimilar in the way they induce other inflammatory mediators. So, as for their second finding, tau concentrations were higher in male athletes who participated in collision sports versus non-collision sports. This suggests that repetitive subconcussive impacts associated with collision sport participation may lead to a greater biological impact than a reported concussion and could have a distinct pathological signature. Tau-laden plaque depositions have been identified in the brains of post-mortem and living former collision sport athletes. More specifically, males who played collision sports were associated with a 62% increase in peripheral tau levels compared to males who participated in non-collision sports. So now on to the conclusion. Male athletes who participate in collision sports are associated with alterations in brain injury related and inflammatory blood biomarkers. Multiple concussions are associated with elevations in MCP1 in female athletes and MCP4 in male athletes. Collision sport participation is responsible for a greater covariance with systemic biomarkers compared to that of concussion history and is specifically associated with increases in tau. So now on to future direction. So more studies are required to understand the biological relevance behind systemic biomarkers in athletes who have sustained repetitive head trauma and those who participate in collision sports in order to be able to fully comprehend the health consequences. There should also be future research on the biological impact caused by elevated chemokine levels after multiple concussions in male and female athletes. There should also be a focus on the variation due to genders. For example, why are there elevated levels of MCP1 in females, whereas there are elevated levels of MCP4 in males? So now on to the critical analysis. So while systemic chemokines have not been assessed chronically after concussions, the results obtained by Mucci-Grosso et al. And Schultz et al. Are consistent with the results obtained by Batista et al., which is the article I'm doing my presentation on, and that there is persistent inflammation months after mild TBI in both animals and humans. TBI stands for traumatic brain injury. As previously mentioned, a major finding was that tau concentrations were much higher in male athletes who participate in collision sports versus non-collision sports. This agrees with the fact that collision sport athletes who have had their brains studied also show significant plaque depositions. And this is my references slide. Thank you very much for allowing me to present. Have a great rest of the day.
Hello, my name is Melody Yang, and I'm a fourth-year undergraduate at the University of Toronto. I'm studying neurosciences and cognitive sciences as a double major, and today I'm going to be presenting to you one of the coolest interviews I've ever done with Claire Fiala, who works at the Diamandis Lab. At the Diamandis Lab, the principal investigator is Dr. Phidias Diamandis, and he's a neuropathologist. And his main interest is creating a convolutional neural network that will be able to detect any sort of neuropathological diseases from looking at histopathology.
I'm Claire Fiala. So I've been working at the Diamandis Lab since September or October-ish. So my official title there is a research analyst. So I'm not a grad student per se, but I work really closely with Phidias, and I help him with a lot of grants. I'm also doing an AI project with him.
A neural network will take any data set in its input layer shown in blue, process it through the hidden layer shown in green, and finally pull all of the results into an output layer shown in red. All of the connections between each layer is called a synapse, and these synapses are weighted statistically depending on how much they are used to classify information. Therefore, synapses have a connection strength. This is how the network learns to classify and process the data set, as the connection strengths are always remembered no matter what new input is fed through the network. As you can see on the left, there is only one hidden layer, and that is the simplest form of a neural network, whereas on the right, there are multiple hidden layers, and is most likely what has been used in Claire's datasets.
Neural networks are pretty good at saying cancer or no cancer, but that even though that's kind of helpful, generally by the time you're like operating, like cutting into someone's head, you probably you probably think that there's a pretty strong likelihood of having cancer. So what's more useful is determining what kind of tumor it is. So the neurosurgeon will take out like a bit of tumor, and then like they'll really quick, or like some tech will really quickly do an H&E stain, which is hematoxylin and eosin, and then they'll call the neuropathologist. So for Diaz, for example, he's a practicing neuropathologist, and they'll be like, hey, look at this, and like tell us your first impressions, so we know what like kind of a broad outline of what we want to do with surgery, like whether we're going to take out a lot or whether we're just going to leave it. So he'll give his like, I guess, his provisional diagnosis, or sorry, he calls it differential diagnosis. So this is my first impression. But then to confirm their diagnosis, he orders like a bunch of molecular tests. So that could be sequencing bits of the tumor, or doing immunostaining or stuff like that to confirm various things. So the way it works essentially is we're using a convolutional neural network to try and identify different types of brain tumor. So I think our network has about 40 types. So
Claire mentions that the lab specifically uses convolutional neural networks, or CNNs. CNNs are specifically designed for visual processing and classification of its data, making it perfect for optical tasks like processing histopathology. Furthermore, unlike basic neural networks, convolutional types will pool results at multiple stages. There are some limitations to the current technology which prevent widespread CNN implementation. However, it does have a bright future in biological AI applications.
What makes like a CNN different from like other AI is because it kind of decides what to focus on. It's not like manual feature detection where we tell it what to look at. The CNN, like, we'll give it a bunch of like 50,000 or so slides, and we'll tell it what the slides are, but we'll let the computer decide what features it's going to look for. So often, like a human, when a human's learning to identify, they're going to be like, okay, well, like look for the swirly line, like look for the swirls or something. I don't know. I don't think that's real neuropathology, but like a computer might look for like this little edge of a line, or like this like weird things that like humans just aren't programmed to look for. But it's more accurate for a computer to choose like what it looks for, like as you can see in the fact that the results have been improving.
That was like sort of like the different layers. I guess each layer is like looking at a different aspect of the shape.
I mean, as you know, with each layer it gets more and more complex, so it looks for more and more like specific things. So it might start in the first layer, it might look for a line, and then the second layer it might look for a diamond, and then in the third layer it might look for a checkerboard. So it's kind of like the function of pooling brings like all the like just brings a whole new level of detail that you couldn't get in ordinary or like in like more traditional networks, because you can take a bunch of units of data and put it to one big piece of data, so you can make more complex orders, not just like a matrix, like a 3x3 matrix. Sorry.
So then, what sorts of advantages would CNN have over, say, a regular differential diagnosis from a pathologist?
What the advantage is, so a lot of the cases that Fidia sees, or like any neuropathologist sees, are pretty routine. So it's kind of like you're wasting your time. So first you have to look at it, and then you have to go through the trouble of ordering the tests, which take a while to come back. So he's thinking, okay, well, it's better. What if we can train like a neural network to like order the test, so I can look at everything like all at once. So this is a lot more efficient.
The efficiency Claire mentions is also reflected in the figure from her lab's paper about developing AI to implement in the clinic. Shown at the bottom here is where CNNs would likely fall in terms of the amount of time it takes to reach a differential diagnosis. You can see that as a patient's diagnosis takes longer time to achieve, the cost of these procedures increases. And, as Claire will discuss next, this attribute of CNNs greatly benefits smaller communities.
A lot of like neuropathology is a really niche thing. So only like the big cities have it. So having a computer that can do it like in the more rural or the secondary like communities, like the more, so that would be really cool because a lot of like even though they might have pathologists there, they're not necessarily neuropathologists. And neuropathology is like a whole world unto itself. So it will mean that like a lot of cases can be it just means that the strain is taken off like the more rural communities or people that can't, communities that can't even afford to hire pathologists because pathologists are expensive.
It's easy to see why smaller communities can benefit from a widely accessible CNN. However, CNNs are not without their own disadvantages. Their inaccuracy may pose a problem in the clinical setting, as Claire mentions that the neural network can get confused. While these errors can be flagged and resolved by a pathologist, it goes to show that we still have a ways to go before diagnoses can be automated.
But it takes like a lot of like computer power and stuff. So even though like they've been theorized for a long time, it's only recently that we've had like the equipment and the technology to be able to actually make it work. And of course, they're still improving.
So it's more like a rediversion of your energy and your assets towards more difficult, lengthy cases, while AI takes over, like automating like something that's, I guess, a lower level in difficulty.
Exactly.
Yeah. So then, a common misconception in the general public would be that AI is trying to replace pathologists. How do you see your lab's research affecting the field of pathology, especially when it comes to integrating AI?
So even though AI is getting good, it's not like good in some really controlled situations. For most applications, it's not good enough to completely replace a pathologist. Because when a pathologist makes a diagnosis, or like a radiologist too, by the same coin, when they make a diagnosis, they don't just look at the picture, off like the image. Often they'll look at like the patient's history, or other tests to get an idea of what's going on. I think though one thing that something that so these technologies, even though like we're working or the lab is working on commercializing them, and they'll probably eventually be used, I think there'll still be a place for pathologists, particularly for reviewing like the more difficult cases. Like some cases, like brain cancer to start with, is really rare. But in some tumors, like you might see, or some types, you might see like once in a lifetime, like one like. So you get really confused. So it gets and there's some tumors that even now that they got all the neuropathologists in Toronto to look at, and like they have no idea what it is. So it gives the neuropathologist more time to focus on the really hard cases, which I think is cool. But it also lets them do research. So it will hopefully, like when a lot of their routine stuff is more automated, it will let them do research too, or focus on like stuff that humans can only provide and computers can't. So right now humans are the main, like are creative, and they can think of like new things or do new research, which I think is really cool. Like so the pathologist can spend less time doing the really routine stuff and more time like thinking through like new innovations, or because the pathology is a big field, or like looking into therapeutics, or like even just working with patients. Like I think that would be a really cool move forward. So,
as Claire says, while CNNs are not foolproof, there is a growing trend in usage. The line graph on the right helps explain why, as it shows that deep learning networks like CNNs have the greatest accuracy compared to other types of networks. On top of that, there is a significant decrease in error over the years, as shown in the bottom figure taken from the lab's paper. The red bar represents the amount of errors made by a human pathologist, compared to the error rates of the most successful CNNs in the ImageNet hackathon. Furthermore, Claire mentions that her AI project has a success rate of about 90%, with the inclusion of over 40 types of brain tumors. As the accuracy of AI gets better, Claire, do you think any other areas other than neuroscience will see more AI research?
AI in medicine is a really hot topic at the moment because it's a really promising area of research that a lot of people are excited about.
Completely. So then, how do you see your field of work progressing in the future?
So much, I mean, in my like humble opinion, so much of the world is becoming more and more interdisciplinary. Like there's a movement against things being so siloed.
And personally, I really agree with Claire. I think AI has the power to be used across all biological disciplines because of its processing. And I want to say a big thank you to the Diamandis Lab, and of course to Claire Fiala herself, whose work was the inspiration for this entire video. I also want to say thank you to Dr. Ju and the HMB 420 team. I hope you enjoyed learning about emerging AI in clinical care as much as I did, and thank you for watching.